Thanks, Jonathan, thanks, Jennifer, and all the organizers for having me. It's been a fantastic first morning, and I really look forward to the rest of the week. So my lab is interested in understanding how the brain produces learned behaviors. And the core idea is that learning is associated with a set of experiences, and these experiences somehow, and I would argue we still don't know how, these experiences drive changes in brain activity that are persistent and are really necessary for an animal to learn tasks that are important for its survival. And in a mouse, and that's the model organism I'm using, this could be remembering how to find food, how to find shelter, avoiding predators, etc. And so, despite this important function, this is really not well understood. And so I'm going to tell you today about our efforts to understand on a sort of mechanistic level how experiences affect, how the brain encodes experiences. And so I say our efforts because this was really, all the experiments I'm going to show you today were performed as part of a team effort when I was still a postdoc in Jeff McGee's lab at Baylor College of Medicine. Okay, so to get at that question, how experiences affect brain activity, we have been studying place cells in hippocampal area CA1. And CA1 is, of course, an area that we know from decades of work is very important for animals to learn tasks and to learn tasks related to space. And so very briefly, what are place cells? So place cells are neurons that fire action potential specifically when the animal is in a particular location of its environment. So this is an example from a rat in an open field. The white line is the trajectory of the animal. Each red dot is an action potential. And you see that this cell fired action potentials, particularly when the animal is in the right upper corner of the arena. Now, in any given environment, about 25 to 60% of CA1 cells are place cells. They have this feature set activity. The other cells are silent. And so the firing fields or place fields, as they are called, tile the entire environment, suggesting at the first approximation, these cells are used for the animal to navigate in space. However, when one looks at a population of cells, it becomes evident that there's more to the story. So this is data from my own recordings. So this is now data from an animal on a linear track. The y-axis is the place cells. The x-axis is the position of the animal on the linear track. And the red color marks for each of these place cells their firing field. And so you can see that, yes, these firing fields tile the entire environment, but there is also a location here at 90 centimeters, where there's clearly an increased density of place fields. And this is the area where the animals that are water restricted and therefore thirsty get a water reward. And so the presence of this so-called reward overrepresentation, which has been found by many other labs before, so this was not our discovery. So the presence of this reward overrepresentation suggests to us that what this representation actually does, it reflects what's important, it reflects what's salient to the animal, it could reflect the experience of the animal. And so we concluded that we could use understanding how this representation forms as kind of a model to understand how experiences actually leave a mark on brain activity. And I should say that actually when you mess with this overrepresentation, at least in this type of task, this type of linear track, the animal will not learn. So the behavior will be affected. So it is important, it is necessary to have this sort of increased place field density to learn the reward location. Okay, so, yeah, sure. Say again. What do you mean uncertainty? Okay. Oh, so sorry. So this is activity here. So what I'm plotting here is the activity of a cell. The redder it is, the higher the cell is active. What you maybe mean is that these place fields are wider than here, and at the end of the talk, you will know why that is. So this has to do with the mechanism, how these place fields are formed. Okay? So just to orient you anatomically, so CA1 neurons are pyramidal cells. They are made up of two dendritic compartments, the perisomatic compartment, which receives intrahippocampal input coming from CA3, and Thomas Odell talked about this. There is the other compartment out here, which is the tuft, which receives long-range excitatory input coming mostly from layer 3 of the entorhinal cortex. Now, we know that these cells here, the CA3 cells, are place cells themselves. So we can assume that inputs that come from these cells will be spatially modulated. Whereas before we started this work, it was much less clear what these layer 3 entorhinal cortex do. There was a description from maybe 2 or 3 papers that said that they were somewhat spatially modulated. So they were described as spatially irregularly tuned. And so the question that I'm going to first discuss is with you, so how do individual cells, how do individual neurons become place cells? So to study this, we use a very simple learning task. I already alluded to that. So animals are head fixed. They are running on a linear treadmill. The treadmill has cues attached to it, usually 3 different set of cues. And so this gives the animal the perception of that they're running maybe down a hallway. And once every lap, they get a reward. And so this is usually at a fixed reward location. And animals readily learn this. So this is from a well-trained animal. So you can see here, this is the mouse's head. This is the head fixation. This is the lick port where the animal gets reward, and we measure the licking. And you can also see sort of the bell passing by with the type of cues that is attached to it that the animal can touch, perhaps see, but probably mostly touch with their whiskers or their paw. And so this is a well-trained mouse. So you can tell by that it's licking really in a restricted area only around the reward location. And it's also has this stereotypical running pattern where it will accelerate after consuming the reward and then slow down as it approaches the reward, consume the reward, and accelerate again. So basically running and licking is what we use as parameters to establish that the animal knows where the reward location is. Okay, so combining this task with in vivo whole cell recordings, we found a few years back that a certain type of burst firing, the complex spike burst firing that also Thomas talked about, is very good, it's very powerful at driving place field formation. And so this is an example of a cell that in labs 5 and 6, and when I talk about a lab, I mean one complete revolution of the belt, or a 100 centimeter running distance. So in labs 5 or 6, the cell was really quiescent. So it fired only spurious action potentials, or only showed this sort of subthreshold activity. Then after firing one of these plateaus, and I will talk about it in a minute what these are, you see that suddenly the cell became really highly firing. It acquired a place field basically in one lab, and these place fields stay for what we can tell for the remainder of the recording. Now these plateau potentials in our hands we call them plateaus because these bursts of action potential are associated with a large dendritic spike that's initiated out in the dendritic tuft of the dendrites. It's called a dendritic plateau potential. And that's why I'll refer to this type of burst firing for the rest of the talk as plateau. These are associated with large amount of calcium that rushes into the cell. And sometimes in cortical literature, they're also called calcium spikes. So this is basically very similar to this calcium spike you might have read about in cortical neurons. Now, because we are in whole cell configuration, we can also play tricks on the cell, so we can influence the membrane potential. And so that's what we did next. We sort of, quote unquote, artificially induced these plateaus, as you can see here. So this is now this is the cell doing it spontaneously. This is now me doing a current injection through my patch pipette and artificially evoking these plateaus. And also in this case, you can see that a place field appeared. Now, we can play this trick anywhere on the track and everywhere it works. So there is not really a privileged position that only there we can evoke these plateaus. If we evoked high frequency action potentials instead of plateau firing, this would not induce place field formation. And through a series sort of analysis and additional experiments that I don't have time to show you, but I can talk to you in the discussion if someone's interested, we determined that this is basically here. What happens here is that the CA3 to CA1 synapses, or a subset of CA3 to CA1 synapses, undergo weight changes. So they undergo synaptic plasticity. Okay, so what did we conclude now from this work? So basically, each CA1 cell receives a heterogeneously barrage of spatially tuned inputs from CA3. So this is shown here by these Gaussians that are all sort of, you should represent these spatially tuned inputs. And so basically, each cell knows about the entire environment in principle. And then after firing a plateau, a cell becomes a place cell because it responds now to a subset of excitatory synaptic weights that have undergone potentiation. The result of this weight change is this subthreshold ramp that then drives the action potential firing in place cells. Okay, so obviously this was very interesting to us, and it seemed very different to what we previously knew about synaptic plasticity. And so to just illustrate that, I want to go back to the data and show you one lab where a plateau happened, and then in the next lab, a place field appeared. This yellow tick mark is basically corresponds to the beginning of the place field in time now. This is in space, this would be in time. And so I want to point out 3 features of this plateau-driven plasticity. So one, it's a one-try learning rule. So as I showed you, only a single plateau is sufficient to drive place field formation, to drive plasticity. 2, it works on the time scale of seconds. So I showed you here, I marked here the beginning of the place field. So this down here is the time in seconds. So you could see that activity underwent potentiation that was active 2 seconds before the plateau itself, right? So there is, it clearly has a large window of plasticity. And 3, there is no causality involved. So inputs undergo potentiation that were so far ahead, active so far ahead of the plateau itself that they had nothing to do with driving the plateau itself. Okay, and so to summarize this now, I want to sort of compare this plateau-driven plasticity, which we call behavioral timescale synaptic plasticity, or BTSP, with sort of the common synaptic rule that we know, which is spike time-independent plasticity. Okay, spike time-independent plasticity, rate changes depend on the correlated activity between pre- and post-synaptic neuron. And this correlated activity needs to happen within a time frame of usually tens of milliseconds. So usually around 50 milliseconds or so. Behavioral timescale plasticity depends on the firing of a dendritic plateau potential. The window is in the seconds range. Exactly, it's 6 seconds, 4 seconds ahead, 2 seconds after the plateau. And we think this window is a consequence of what has been mentioned now several times before, where we have a sort of a correlation between the plateau and what we think is a eligibility trace. And this eligibility trace, so I put a question mark there because we don't know what it is. It's still something we are looking for. Should be some sort of filtered, should be depend on the synapse being active, should be synapse specific, should depend on the synapse being active, and also work on this time scale of seconds. Okay, so this is what we know, but we don't really know what it is for the moment. Okay, so this asymmetry in the plasticity window has one interesting functional consequence, namely that place fields that were driven by the plateau, by BTSP, actually precede the plateau itself. So as you can see here, so we have a plateau happening, and then the place field firing, most of the place field firing happens before. On average in our data sets is between 10 and 20 centimeters. So what actually BTSP does, it produces not place fields that exactly are at the same location, but place fields that are predictive, that sort of predict whatever drove the plateau itself. So this is one interesting functional consequence. The other feature that also came up this morning already, is that it's bidirectional. So this came from experiments. So this is really work by a former postdoc in Jeff's lab, Aaron, who is a computational neuroscientist now. So we had this finding that if we have a cell that has a place field in one location, and we induced it, for example, and then if we induced it in a different location, the place field would translocate. So this is a firing map. So the redder the color, the higher the firing field. So you can see that initially the place field formed here, and then it sort of translocated ahead because here is the second induction site. So based on this data, Aaron came up with this weight-dependent rule that was mentioned this morning, where actually whether a synapse will undergo potentiation or depotentiation will depend on 2 factors. One is closeness to the plateau potential itself, and 2 is the initial weight. So this is basically the summary of this work. You can see that if a presynaptic input happens directly at the plateau itself, so this would be time point 0, you can see that most of it will undergo potentiation, but if it really has a high initial weight from the y-axis, actually there will be some conditions where this input will actually undergo depotentiation. And so with this sort of weight-dependent rule, we can then explain these translocations here. Okay, so these are sort of the features I mentioned to you. I didn't have time to show you the data, but the pharmacology, it's basically very similar to SDP. So it's also dependent on NMDA receptor activity and on calcium channel antagonists, because the plateaus are calcium spikes. So if you block R-type calcium channels, voltage-gated calcium channels, you will block plateau initiation, and this will block BTSP. Okay. If there are any questions, now would be a good time to stop. Yeah, Rob.
It's amazing work. So how homogeneous are plateaus? So what do you think is really critical for the plasticity? Is it the calcium spike and the plateau depolarization? Is it the spikes riding on top, or are they pretty negligible, you think?
I see. So how homogeneous are plateaus in a cell?
Yeah, for example, in the first example you showed, you induced a plateau and nothing happened. And then a few trials later, you induced another plateau and the place field formed.
Okay, so this is, this is a great nuance that I usually, let me just go into it's a great question. So here, this is really the spontaneous plateau. Okay, so this is about 300 milliseconds long. So here, we do it artificially. And so for the artificial induction, we usually, the protocol is 5 to 6 labs, always in the same location, a high current injection. Now, in some cells, something will happen immediately. In others, we really need the 5 trials. And so it's a speculation, but we think it just shows that there is some additional factor missing that is present here. Some sort of novelty in neuromodulation, indicating novelty, priming the dendrites, putting the dendrites in the right excitability state to immediately drive a really strong calcium spike, for example. So in the artificial induction, if you do it 5 times, it always works, but the response varies from cell to cell, because probably there is some additional things. Also, inhibition could be a factor, right? So there could be additional factors missing that in the spontaneous plateau case makes drive plasticity right away. Does this make sense? And then another nuance which I find very interesting is that cells will have so basically, there are hundreds of milliseconds long, but there are also shorter ones, 50 milliseconds long. And we don't have enough data to go after that, but the assumption is that these are the ones that drive plasticity. The shorter ones are what we call the coding plateaus. So they code, mixed selectivity, some sort of difference to the downstream cell, but they don't drive plasticity by itself. And you will find those in the place field afterwards as well.
And why the -4 to +2 second long window? What sets the time scale for that, if the plateau itself is a second or so?
I'm not sure I understand the question. So what do you mean? So this is not a second. This is maybe 300 or 200 milliseconds.
Oh, okay, yeah.
Yeah.
I mean, you highlighted the stereotypy of the overall time window for BTSP, right?
Yes.
So what sets the extent of that time window?
So we think this is what the eligibility trace. This is, if we model that, that's a biochemical, this is what's set by the eligibility trace. And by the way, there is now a new preprint out from Jeff's lab. They look in CA3. The window is actually a little different. And probably because the eligibility trace, the biochemical signal is different in different cell types. Yes?
So what triggers this? If you record for 2 cells at the same time, do you see the potential at the same time?
Okay, so this is basically what the second part of the talk is about. What triggers this is an input from the entorhinal cortex. So we know this on the population level. Now, what happens in exactly this moment to the mouse that it has this, we don't know. We can just speculate. It's probably, I don't know, anthropomorphizing here, but it could be, something special needs to be remembered about this moment, but we don't know what happened exactly in that same cell. Because there will be, as you said, a neighboring cell that will not undergo BTSP. Yes?
Yeah, so just a difference to, what is different about the current injection to induce a plateau or to induce just a number of spikes?
So here we induce plateaus. You see this depolarizing wave running on.
Yeah, but what is different about the current that you inject to get a plateau instead of
versus action potentials?
Yeah, it's just the amplitude.
Just the amount. Yeah. So the amount is lower, but the frequency of action potential that is riding was the same. Okay, so we also was 200 hertz action potentials. It was just less, so that there was none of this depolarizing wave, which is the expression of the calcium spike at the soma. Yes?
You should show your translocation plot. So this one.
Sorry, what plot there?
The one we showed, you talked about translocation of that one. So there, the place field seems very coarse compared to the place fields you were showing, for instance, right at the beginning.
Yeah.
So is that because it is yet to refine? Also, we know that place fields can have more than one field.
So I left this whole part out. So basically, the 6 seconds window has the consequence that depending on the velocity of the animal, when the plateau happens, the place field will be wide or narrow. So this was probably a location where the animal was running very fast in space, meaning that in the 6 seconds, it crosses a much wider space. So in the same time window, many more inputs will be, or many more eligibility traits will be there to be potentiated. Now, if we would do this, you see here, this is narrower, and I show you later on another place field with calcium imaging, but it's basically around the reward, and this was the observation here, right? The fields around the reward are very narrow. That's because the animal there is running at low speed, because it consumes the reward, fields will be narrow.
It's crystallized at the very first point. The whole width is crystallized, it never changes. It doesn't refine.
In our hands, no. In our hands, no.
Thank you.
Maybe we can continue and
sure.
get some more questions.
I like the questions. Okay. Okay, so we were, right, so also happy to answer any more questions in the panel discussion. So because of all these features, PTSD really could encode a single experience, right? Because it is a one-trial learning rule, so it really needs just one event to drive these plateaus. And so the next question that we then wanted to ask is, so is BTSBs actually, does it allow experiences to shape representations in CA1? And plateaus could be sort of a teaching signal, telling the cell, as I mentioned, something interesting has happened, now form a place field. So to get at that question now, single cell recordings are not really a good method. So we switched to photon calcium imaging now. So this is just an example, part of my field of view. So these are all somata of CA1 pyramidal cells that are labeled with the genetically encoded calcium indicator GKM6F. And so you can hopefully appreciate that this allows us to monitor the activity of many cells at the same time. Here I just show you 9 representative place fields that sort of tile the environment. They're randomly chosen. So this is just to give you an impression of the signal to noise ratio that we achieve. Okay, so the task is now a little bit different. So I now really try to catch the cells when the environment is completely novel. So I have these 2 phases, training, and the first day of imaging happens on a totally blank belt, random reward locations. And then on day 1, I introduce them to the first time to the fixed reward location and to the fixed reward location and the belt with the cues, as I've shown you before. And so the learning, to show you that these animals really learn that, this is the representative licking behavior from one animal. Each of these ticks is a lick. Y-axis is the lapse, x-axis is the position. So this is from one session. And you see here in these conditions, the animal basically licks everywhere because it doesn't know when the reward comes out. But then on this fixed day reward conditions, with basically within 30 laps or so, the animal learns to restrict its licking to a tighter and tighter zone, showing to us that really this is a simple learning task that they can learn within a single session where the reward location is. Now, looking at the calcium activity at the representation, so this I've showed you before. So basically, we can see in one session this reward over representation form. I should also say that there's basically no overlap between these 2 representations. So these are cells recorded on day 0 and cells recorded on day 1, but from the same animals, the same field of views. But there is very nearly no overlap. So we can basically watch the representation form under the microscope as the animal learns this task. And this is just a quantification. So I basically just count the number of fields as a function of location. And here it's uniform, as you can see. And here is this reward over representation forming. Now this is what happens on the single cells. So these are 3 representative place cells that are recorded on day 1 as the animal learns. And here you can hopefully appreciate now the difference in the width. And that's exactly what I mentioned. The reward here is at 90 centimeters. So for example, this place field will be very narrow. This place field will be higher because here, just in this lab, it was formed when the animal was running at higher speed. But you can also see, hopefully, that it's very reminiscent of BTSP. The cell is not active, and then suddenly it's active. So in one trial, a place field forms. Plus, we see also this predictive character that I mentioned before, that the place field that is formed by the plateau actually precedes the plateau itself. And I don't know if I have it. Oh yeah, and I also did the pharmacology. So I applied an NMDA receptor antagonist and SNX, an R-type voltage-gated calcium channel, a plateau blocker. And in this case, two things happen. So the number of place fields goes down. Usually in our case, it's 25%. Now it's only 16 or 12% of the cells are actually place cells. And overall, there is not as much of a reward overrepresentation formed. And generally, the representation is more uniform. So this is what we conclude from this work, from this initial part, is basically you have these two inputs. You have BTSP on the CA3-1 to CA1 synapses, and this creates experience-dependent and predictive representations in CA1. Now, the last question is, what drives the plateaus? And basically EC3, the other big input that goes to the tuft, is a good candidate for that, because we know from previous in vitro and in vivo work that if you silence EC3, plateau probability will go down. Okay, and so I wanted to know whether basically also EC3, if you block EC3, place field formation is decreased. So this is what I did. So I first silenced EC3 inputs as the animal does the task. So we used a EC3-specific driver line that we got from the Tunagawa lab. I injected a retrograde virus in CA1 expressing the optogenetic silencer ARCT, and I expressed GCAM6F just in CA1 neurons. And so, this here you can see the ARCT expressing axons nicely in the tuft area where these cells receive the EC3 input. These are the GCAM cells, and these are the ARCT tomato cells in EC, where I also implant the fiber to activate ARCT. And so this is then the result. So here I just silence EC3 where the red bar is, so basically only around the reward. And you see in the T-tomato control, the reward overrepresentation forms normally, whereas here this is reduced, and this is the quantification for that. So first, ARCT activation avoids place field formation, or seems to silence. Yeah.
So for both this and the pharmacology you just showed, what happens to the behavior? They're looking for specific locations.
Yeah, no change in behavior in both. And I think this is by, so for the pharmacology, and I'm not surprised for the I'm not surprised for both. So this is just unilateral silencing, and the pharmacology is really just locally where I image. And this was by design, right? Because the whole circuit is a loop. And EC3 is also connected to layer 2, which is basically the main input to the whole hippocampus. So we wanted to, by design, limit the amount of silenced cells. That's why I chose this retrograde approach, so that it's really only the EC3 cells that connect to these cells or connect roughly to the area where I image, that they are expressing ARCT. So we didn't see any behavioral deficits. My expression, so I haven't done that, but my expectation would be that it goes down, because as I mentioned, Attila Losonci has done experiments where he decreases the overrepresentation, basically like this. You do another manipulation of interneurons, and there he sees a behavioral deficit. So I would expect a behavioral deficit.
Sorry, do you mind drawing one slide before?
So this is.
When you have the representation of individual cells.
Sorry, say again.
When you have your representation of individual cells, the middle plot looked like there was a satiety effect. Your representation was stronger on the beginning of the first trials.
This one, you mean?
Yeah. Have you ever looked at what happens to the representation when the animal is not thirsty?
We have not done. I mean, we have not done that because I think the animal wouldn't run. So, I mean, we do a mild water restriction, right? They do maybe lose 10% of their body weight. So I don't think they are, super, super thirsty. We have never done it in a non-thirsty animal because this is just our way of motivating them to do the task. So I don't know what would happen. My expectation would be it would still work, but I don't know. There are many questions. Can I just maybe finish and then we do the questions after? Okay, so we silence EC3, and so we see a decrease in place field formation. So we wanted to know what do these cells actually encode. And so this I maybe skip. So this is just showing you that we can image those EC3 axons. So basically we put an imaging plane here now in the stratum lacunoso moleculare, and that way we can image the axons that go to these cells. And so this is how what these EC3 cells do. So these are 3 cells, labs, position, and so you see they are clearly not like place cells, right? Their reliability is very low. Their spatial selectivity is not that good, and so these are really 3 representative place fields. And so if I to illustrate a little more, I plot them now like this. So I split the data up into odd labs and even labs, and then create averages of both these 2 parts of the data set. And then I make heat maps based on the odd labs, sort them based on the odd labs, and then I sort the even labs based on this. So what you can see is basically, if I plot now here the peak location in the even labs versus the peak location in the odd labs, you see there's basically 2 separate populations maybe. So some are really very uncorrelated with each other. So these would be these that are outside of the unity line, but there is a population of cells that fall on this unity line. This would be sort of the cells that at least preserve some spatial selectivity if we split up the data sets into 2. If I now ask what's the spatial distribution of these cells on the unity line, it's basically uniform. So this was, in the first approximation, quite disappointing, because I guess one of the hypotheses would be that there's just more EC3 input around the reward. That's why we have the highest amount of place field formation around the reward and the reward overrepresentation. But that doesn't seem to be the case. What we maybe thought this could mean is that what these EC3 cells encode is a reflection of the uniform Q distribution on the belt. So the belt has Qs everywhere, so maybe that's what these cells encode, and I will come back in 2 slides to that idea again. But so how could this then drive the representation that we observe? So to get at that, we went to modeling, and basically made a very simple Markov chain model where we have 10,000 CA1 neurons, 2,000 EC3 axons, and these EC3 axons, the transition probabilities for the Markov chain model are selected in such a way that they recapitulate well what we see in terms of the axon activity I showed you before. Okay, and so now we select for each of these 10,000 CA1 neurons, we randomly select 100 EC3 axons and sum up their activity over time. And so this is how it looks like. So these are 5 different, this is the activity that 5 different CA1 neurons would see if we just sum up this activity sort of linearly. We just sum it up. And so based on this, then we can ask, so what is the probability of seeing a threshold crossing event, which would be a plateau evoking event? And the threshold is chosen in such a way that we have these events in about 25% of the C1 neurons, which reflects about what we see in our population. And so then we can do 2 cases, one where we impose a constant velocity or constant dwell time. So now the animal is running at constant speed everywhere along the track. In this case, we don't see really, we see a uniform distribution in space for these, plateau evoking events. However, if we impose the real velocity now, which means the animal slows down around the reward and then speeds up again, this is just the inverse of the velocity, so that's why it goes up. You see that in this case now we can explain why there is more place field formation around the reward. There's place field formation everywhere, but especially around the reward. And so from this, we then can conclude that this activity profile is consistent in principle with an instructive signal, but it has different components. So it has the activity and it uses the behavior to impose a non-uniformity. Now, to really test this idea, we did another experiment. I designed a different environment. Now, you see, this is the normal environment with the cues, 1 reward location. But now I had a different environment with an empty belt and just 2 cues, 1 reward predictive cue and the reward itself. And so you see now in blue, the EC3 target signal looks very different. It now actually increases not around the reward anymore, but it increases around the light. We don't know why, but this is just what happens. If we now put this target signal into our model, we would predict an overrepresentation of the light cue, not of the reward now. And this is exactly what happens. So basically what we can show here is that this instructive signal is is environment specific. And actually, if we change it, CA1 representation also changes accordingly, which is, I think, a really nice result. Okay, so final question. So is this instructive signal an arrow or a target? The difference being that an arrow signal would decrease with learning, or a target would stay constant. And so this is our attempt to get at that. So basically, I'm showing you just now some population activity for CA1 and EC3, first CA1. So this is for the first 10 labs and labs 51 to 60. So you see, and this is in this environment B with the light Q. So you see initially it's very uniform and very low, the activity level, but then after learning, because more place fields were formed, were added, you see then essentially the overall level goes up. Plus we have the overrepresentation of the light here at 40 centimeters. If I now plot on top the EC3 target signal, the EC3 signal, you see that basically it doesn't change much, right? The level stays the same. Overall, the shape stays the same. And so this is also true when I now plot sort of this level here across 80 labs, across the first 80 labs. So it stays more or less constant. Whereas if I plot the correlation between the blue and the red, you see that it sort of initially is very different, and then it approaches this level. Okay, so this is then sort of the model we have right now of BTSP. So we have a target signal coming in from EC3. In the apical dendrites, you have a mismatch calculation going on between that target signal and a signal that reflects the actual state of the CA1 representation. We don't know what that is. The first candidate mechanism we're thinking about is feedback inhibition through SST into neurons, because there's a class of interneurons that actually receives feedback input from CA1 and then targets specifically the tough dendrites. So if there's a mismatch between this signal and this signal, you have an arrow signal, you have an excess excitation, an arrow signal, which drives the plateau. The plateau changes via BTSP, the actual state of the representation, and so this computation begins anew. Okay, so this is the current model. Of course, many open questions, happy to discuss those. As I said, all of this work was performed as part of a team effort in Jeff McGee's lab. Now I have my own group at Brandeis. Super fun group to work with, and thank you for your attention.
Thanks, that was really great. I have a couple of questions about the interaction between time and space. And so I guess part of this you answered during the talk, but I guess one part of it is there any role for theta cycles in defining that relationship? Because those also compress or expand depending on the running speed of the animal, as I understand it. And then I guess
future cycles, you mean future labs?
Sorry, theta the theta oscillation.
Okay. Yeah, great question. I mean, the 6-second plasticity window clearly crosses theta cycles. So in the current model, it's not incorporated. However, we are not looking at the single spike level. So there might be some, additional refinement going on through some theta cycle, theta oscillation mediated process. I will say that the plateaus do have a theta preference because the entorhinal cortex comes in a certain theta. So that comes in at the peak of the theta cycle. So this is where this comes in. Right now in the model, we don't incorporate it.
But you don't think the window size would be measured in cycles rather than being measured in time? I
mean, so far the data fits really well if we go in time in seconds. So this certainly crosses several theta cycles.
And then I guess the other...
Which doesn't exclude, right, that this has a role maybe in how the downstream neuron reads it out or something, right? So this is possible.
And then the other question I think also relates to Peter's. So the mapping between time and space isn't constant, right? The animal will run at different speeds at different times as it goes through. So, I mean, I guess under this model, it's the first time that the place field is established that defines the width. And if it then runs faster or slower, the width of the place field, therefore the firing time will will compress or expand,
Yes,
and the
so
width of the base field would stay the same.
Exactly.
Is that seem to be true?
Yes, so we in the initial description of this window there, we did this. We exactly, shuffled the labs we would ask to look at the width, and this is exactly true. So it's only the first lab that matters.
Hi, fantastic work. Two questions. One is very naive. So when you showed the effects of these manipulations, various manipulations, pharmacology, blocking the input from EC, what you showed is that the effect of these manipulations was that it abolished the preponderance of place fields needed, but there was still the normal level of place fields. So do you think that those place fields are based on, the establishment of those place fields is based on a different mechanism? Or is there something very special about these place fields, these extra place fields that you're then blocking with these manipulations? Okay,
so first in the pharmacology, where I basically silence everywhere along the track, the place field number does go down, right? It's not, and presumably, so the assumption is there, it's everywhere, but I just point out the raw representation because that's where the highest effect is. And so I do think that probably not every place field that we're seeing, even in this novel environment, does require BTSP in this environment, because as every cell has some previous experiences, there will be a pre-existing weight matrix in each cell that CA3 can use, right? That CA3 can connect to. So if there is cells that have an increased weight from pre-existing experiences, and CA3 cells will activate those increased weight cells, the cell will have a place field. So, right, so there is probably a baseline in each cell. Maybe the 10%, 15% is kind of maybe this is sort of a conservative estimate of cells that don't require BTSP in any given environment. But my bias would be that they had BTSP events in the past, right? In another environment, it's not the first time the animal is on a treadmill under the microscope, right? It's just the first time it sees this belt with the cues on it. Does this make sense? Or
yeah, it does. Thanks. The other question was also a follow-up to Peter's earlier question about the refinement of place fields. So I was wondering how you see, how you square your results with kind of more, earlier or more classical results from Mang Mehta about place field expansion and shift over repeated exposure.
Okay, so So I was a little hesitant to answer that because the only time we can really be absolutely sure that we see plateaus is with the whole cell recordings. And this gives us a limited recording time, right? With the calcium imaging, we do see occasional, sort of this type of predictive movement overlaps that Mayank sees, but we can't know, are these plateaus? Did additional plateau happen? That's why the shift happened. Or is it really, sort of the SCTP-like mechanism he suggested? Now, with the new voltage indicators, we will be able to answer that really unequivocally. But with the calcium imaging, it's just a little hard for us to see because there could be additional plateaus there. The only time I'm really sure is when there was nothing and then there is something, right? Does this make... So that's why I think his results could be explained by additional plateaus happening, and he with his external recordings can't discriminate it as well.
Right. The curious thing is, though, that his effect, I think, reset on every day or every session. That was my understanding.
Okay,
So it was present over consecutive laps of the same day, but then the next day it would kind of reset. That was my understanding.
I mean, what would you conclude from that?
Well, I'm just wondering what you would, how you would imagine that might work with any of the mechanisms that you just mentioned.
I mean, the other thing that we haven't discussed at all is the possibility that maybe there is also plasticity on the Taft synapses, right? That encodes certain things. So, you could imagine maybe there is some pre-existing weight changes also in the Taft synapses, then would cells give higher propensity the next day in a less novel experience to drive plateaus as well. But we haven't explored that yet. So this is really,
Thanks very much.
In relation to our conversation earlier about homostatic plasticity, you have this beautiful example where you have one place field, you induce another place field, and the other place field immediately goes away, implying this heterosynaptic LTD that Cliff was talking about. But what I understand about the rule is that you have a 5-second window, more or less, for the LTD. So is the prediction that you could create a cell that has 2 place fields if you slow down the speed of the wheel enough to outdo that lap? And have you tried doing that? Because this really addresses what we were talking about earlier.
Yes, so if we can get 2 place fields that don't influence each other, just it needs to be outside of this window. So just our belt is 180 centimeters. You know, the animal runs 40 centimeters a second, so it's very hard to do that in our setup, but yes.
Have you seen that, or just?
We have seen it, you know, anecdotally.
Very cool.
And I think it's also, the example is in the paper, I think.
We'll take 2 more questions and then we'll move on to the next talk. We can continue the discussion later.
Okay, you might have said this. You showed the really nice tiling of the environment by place fields, and then you showed that when they appear, some appear on lap 10, some appear on lap 50. Is there structure to, in terms of the space, how they appeared? Are they sequential? Do they first map everything and then increase the resolution?
So our very crude attempt to get at that is that we just split up the session into 5 labs. So we have 100 labs, we split it up into 5 sections, 20 labs long, and then we ask what's the distribution of the fields formed in this section, and there doesn't seem to be a structure. It's always the same structure, just the amount changes, right? So I didn't have time to show this, but basically it's kind of an asymptotic distribution. So initially you have the most place fields appearing, and it gets less and less and less, sort of consistent with this idea that the plateau is an error. Does that make sense? But spatially, it's always the same distribution. Okay, just the numbers change.
Thanks so much for the talk. So it's kind of a related question. You know, the plasticity you see is one shot, but the behavior is not, right? And so have you found kind of an interpretable relationship between how place fields come online and how that stabilizes the behavior?
I mean, there is some sort of, you know, there's a correlation between so if we give as a readout the fraction of licks outside of the reward, out of the reward zone, which kind of should reflect the refinement of the licking, and then we ask, so how correlate, and then we ask we split it up in sections again, and then correlate the end with the first second. There is sort of an inverse relationship. But this is just correlation at that point. I think what you would want to do is you would want to screw up the plasticity in sort of an interesting way, and then, you know, ask, you know, does this change the behavior or something like that? There is this correlation. I think also the fact that if you don't get the reward representation, behavior is screwed up, shows that there must be something. But I think what I would like to do is some sort of, I don't know, change the time course in an interesting way, or you know, make it less asymmetric, and then see there is a change in the behavior. I think that would be a really cool experiment.
Let's thank Christine again.